Nucleic acid structure. Nucleic acids, DNA and RNA, are the molecules that store genetic information and instruct the cell in creating proteins. Their scientific names are deoxyribonucleic acid and ribonucleic acid. Nucleic acids are made up of carbon, hydrogen, oxygen, nitrogen, and phosphorus. DNA and RNA are made up of chains of nucleotides. Therefore, two nucleotides bonded together is a dinucleotide, and the general name for polymers of nucleotides is polynucleotides. Both RNA and DNA can be found in the nucleus of eukaryotic cells, but the term nucleic acids can be misleading. Prokaryotic cells have both DNA and RNA, though they have no nucleus. RNA is found in the nucleus and the cytoplasm of eukaryotic cells. DNA was first discovered in 1869 by Friedrich Miescher, who named the substance nuclein because he found it in the nuclei of white blood cells. The term was later changed to nucleic acid. Discovering the famous double helix of DNA was a major scientific breakthrough. In 1953, Watson and Crick built the model of DNA below, based on data provided by Franklin, Chargaff, Meissner, and others. DNA stores genetic information that is used for structure, daily functions, and for heredity. Genes are made of DNA and encode all the molecules required by an organism. DNA is the main component of chromosomes. Chromosomes are copied and passed on from parents to offspring during reproduction. There are three types of RNA: messenger RNA, or mRNA, transfer RNA, or tRNA, and ribosomal RNA, or rRNA. Together, they transfer the information stored in DNA to the metabolic machinery of the cell and help to carry out and control all aspects of protein synthesis. Nucleic acids are composed of complex monomers called nucleotides. Nucleotides are made up of three chemical groups: a pentose sugar, a phosphate, and a nitrogenous base, also called base. Because nucleotides are complex, they're often represented with a rectangle, pentagon, circle for simplicity. The methyl group is nonpolar and generally non-reactive. When added onto a molecule with a covalent bond, it changes the way a molecule interacts with other molecules and acts as a signal. It's found in all classes of biological molecules, a common component of DNA and proteins. Cytosines are a component of DNA. Methylation of cytosines makes genes inactive. RNA contains the pentose sugar ribose, while DNA contains deoxyribose. Nucleic acids are the only class of biomolecules that has phosphates as part of their backbone. The presence of phosphate groups gives nucleic acids a negative charge. The phosphate is attached to the C5 of the pentose in a nucleotide. DNA contains the bases adenine, guanine, cytosine, and thymine. RNA contains the bases adenine, guanine, cytosine, and uracil. The difference is that DNA uses thymine rather than uracil. Bases composed of two-ringed structures are known as purines. Bases composed of one-ring structures are known as pyrimidines. Both DNA and RNA contain two purines and two pyrimidines. The bonds between nucleotides are called phosphodiester bonds. Like bonds between monosaccharides and amino acids, nucleotides are joined by dehydration synthesis reactions. The dehydration synthesis reaction joins the phosphate of one nucleotide to the C3' carbon of the pentose in the next nucleotide. The phosphodiester bond is C5' OPOC3'. RNA is a single strand of ribose-containing nucleotides connected by phosphodiester bonds. All four nucleotides are shown: C, G, A, and U. Cytosine, or C, is outlined in red. The new covalent bonds produced by dehydration synthesis reactions are indicated by arrows. One phosphodiester bond has been highlighted in yellow. Nucleic acids have sugar-phosphate backbones. A continuous line drawn through the RNA molecule's sugars and phosphate groups traces the backbone, as highlighted in yellow. The nitrogenous bases extend away from the backbone. The sequence of nitrogenous bases is the coding information for life.